Cognitive Modeling. He is a senior member of IEEE, member of ACM's Society for Neuroscience and Cognitive Science Society. Today, he will be talking about graph diffusion models of the brain functions. Over to you, sir.
Right, thank you, Azra, and thank you, Srinivas, and all the lab members who have organized this very nice. Happy to be part of this. Today, I'll talk about a kind of models based on graph diffusion for modeling brain function. So, a brief slide on functional MRI. As you all perhaps know, that the left-hand side is an MRI scanner, typical MRI scanner with a magnet and a table on which the subject lies down, and then this table is raised and inserted, wheeled into the bore of the magnet to obtain recordings, non-invasive recordings of brain function. Right, what you see on the right hand side is typical response that is collected. This is called the bold response, blood oxygen level dependent signal. So functional MRI or MRI captures the oxygenation level changes in the blood, and over time how it is happening, and the different colors refer to different areas. So we are simultaneously measuring typical nowadays with 3-second repetition time. That means you can scan the whole brain in 3 seconds. You get close to 1,70,000 measurements every 3 seconds all over the brain. Right? You have this time series, and you can collect this over time. And the recent times, the last 2 decades or decade and a half, the exciting discovery is that even when the brain is not really engaged in a task, right, resting brain also is quite active. And so this is the famous quotation, resting brain never rests. So what is this resting state functional magnetic resonance imaging? This is a spontaneous activity, low frequency fluctuations in the bold signal when subjects are asked to close their eyes and review the day, or in another mode of experiments, they are asked to focus on a fixation point and basically think nothing, right? Just keep focusing on that for a typical session lasts for 8 to 10 minutes or 12 minutes. So you are looking at the spontaneous activity, and this whole excitement started by Bharat Biswal at New Jersey Institute of Technology, who has actually reported these kind of spontaneous fluctuations in the resting state in the motor cortex. Of course, it took a long time for people to accept this, but eventually, in the beginnings of 2000, 2003, 4, this has become a rage. That resting state activity and various brain synchronization, brain regional synchronizations that are observed. And so this phenomenon is well known actually in, if you look at the frequency spectrum, is the power of the signal, the blood oxygenation level signal. If you decompose that and look at the power distribution across frequencies, what we are talking about is these low frequency fluctuations below 0.1 hertz is what corresponds to the resting state activity, the slow fluctuations in the brain when subject is just resting. So this resting state function reveals the intrinsic functional connectivity of the brain. And people have studied this. One way that people have approached the standard old method is to take seeds in different regions of interest. For example, there is a seed in the medial parietal cortex precuneus, and suppose you look at what is the response at this location when the subject is resting. Suppose the bold time series is some fluctuations like this over time. Now what you do is you now look at how this is correlated with other regions. So you take an ROI in the left occipital or temporal area, then you see that the blue is this time series, and you look at the correlation of this, there's 80% correlation. And this is just a schematic to give you an idea what is this approach called seed-based correlation approach. You take another ROI in the right hemisphere, it has a different bold signal, and the correlation is some value. And similarly, you take a frontal point and look at the correlation between the seed and this region, right? And like this, you can go through different regions of interest and different seeds, and you can actually populate the whole, look at the whole functional connectivity of the brain. And you can give some sort of color coding, and you get a resting state functional connectivity sort of map for this, where this color coding with yellow refers to very high correlation, and red is medium, and so on. Blue is low correlation. Now, people have studied this, and this seed-based approach is not the only approach to get resting state functional connectivity. People have used, instead of this sort of roughly model-based approach to look at these different regions that are cooperating among themselves, you can also actually extract using model-free methods like ICA, independent component analysis. You can actually extract the spatially distinct networks which are correlated together, right? There are several approaches to get to these resting state so-called networks. What are these? These are synchronous activations among regions that are spatially distinct and in the absence of any task or stimulus. Several such networks have been identified. This is a typical A to G. There are different networks, 7 networks that are depicted. They are given names because of certain characteristics. One most important of them is what is called the default mode network, A. What is shown here is the lateral view of the brain and the medial view of the brain, and the regional correlations on a scale of 0 to 1 is high correlation is yellow and lower correlation toward the red part of this kind of color coding. Now, what you see areas in posterior cingulate cortex, medial frontal cortex, and hippocampus. These are the major players that are active together, and this sort of network, this sort of regional cooperation and working together is seen whenever there is no task. Brain is not engaged in any task. This default mode network lights up. Sometimes this is also called the task negative network. And there are other networks, somatomotor network, for example, B, which the lateral and the medial view, you see that this motor and sensory strip is active in this, both motor, premotor, and sensory areas are active in this network. And there are other networks that are identified, attentional networks, executive control networks, language networks, visual networks. The names of these are somewhat different now. Since they are now called salience network, these attentional, ventral attentional networks are called salience networks. And the control networks are called the executive networks, and so on. But many people, this finding has been replicated in several labs in several kinds of patients and ages. People have seen these resting state networks. So one question in this context that comes up, let me define two terms, what is called structural connectivity, SC, and functional connectivity, FC. So you take the brain and there are two kinds of measurements broadly possible. On the left-hand side, you have anatomical measurements. So you can make a diffusion tensor imaging, DTI imaging, which gives you the fiber tracts that course through the brain and connect different regions of the brain. And you can look at these strength of these or the number of fibers that connect different regions, regional pairs, and you can stack them together in a matrix. This is the one axis. This is the number of regions of the brain. Suppose you parcellate this whole brain into 68 regions, 30, let's say 34 on either side of the brain. You have the 68 by 68 matrix that gives the pairwise fiber strengths that are connecting each of these pairs of regions. This kind of matrix is called an SC matrix or a structural connectivity matrix. On the right hand side, what you see is the functional connectivity matrix. So we just talked about the bold time series that you can measure. You aggregate these over the 68 regions, and then you look at 68 by 68 pairwise correlations of these average time series, and you integrate all these correlation coefficients or other measures of time series similarity. You can use any of the measures that are available for time series correlations, and you can put them, summarize all of them in a matrix. Again, this could be a 68 by 68 functional connectivity matrix. Now, what has been observed empirically, studying these structural connectivity, functional connectivity, is that there is more functional connectivity than can be explained directly from the structure. So in other words, there are correlations that you observe for whom there is no direct anatomical connection. And so the question that arises is, how does the functional connectivity arise from the structural connectivity? In some sense, this has been a major question, that this structure function relationship characterizing this is is an important question in both resting state and task based functional MRI measurements. So the other question is, if in many cases it's also observed that you have the functional, you have the bold time series, but you don't have the structural connectivity. So another corollary question is, can we recover the structure from the function? Remember that there is more function than structure, so that that this recovery is not trivial, right? So to address this question, what we thought, our colleagues and I, our students, we thought looking at diffusion phenomenon over graphs. So this can be looked at as a graph with 68 nodes, 34 on either side of the brain. And now we have a graph, and the connectivity is defined by the structural empirical observations about how many fibers connect each of these pairs of regions. Right now, you have a weighted graph on which there is a function that is observed. Is it possible to model using diffusion phenomenon on the graph? And this is work jointly done at IIIT with colleagues. Avinash Sharma is in Center for Visual Information Technology, and Govinda is the student who did this work. Deepanjan at that time was at an IIIT and moved to NBRC subsequently. So what we thought is that you can consider that a node in this graph is the source of heat, and heat spreads over the graph. And this comes from the heat diffusion kind of ideas inspired from that. This heat diffuses to reach other nodes through the edges. These edges are defined from the structural connectivity. And once the heat reaches other nodes, they also start diffusing to their neighbors, and so that this spreads over the whole graph. And temperatures that initially they increase at the nodes, but eventually they settle down into some sort of a distribution over this graph. Now, this can be modeled by a linear differential equation of this kind, where u of t is the regional activity, and there is a spread of this activity in this sort of exponential fashion. The solution of this is, for example, that u of t is e raised to minus beta lt u0, the initial heat distribution and how it diffuses over time. Now, this is a special sort of operator. Here, L here refers to the morphology of the graph in the form of graph Laplacian. This is defined as, L is defined as d minus w, where d is the degree matrix, so degree of each node, how many edges are each of those nodes have, minus the adjacency matrix, how many the adjacency matrix defines the graph edges of this graph. So Laplacian is this, it depicts the structure of the graph, and minus this beta t is the scale of diffusion. Now, this is a diagrammatic representation for schematic representation. If here the node sizes refer to degrees of the nodes, that means if you have more connections, you are bigger size. And the color coding here depicts the amount of heat that has diffused. If you take this as the source or the seed, and then you look at how this the heat that is started from here, how it diffuses over this structural connectivity graph over the entire graph or network, right? So however, in this is a single source that is diffusing. However, in the brain, you can think of multiple diffusions happening over this graph. And this can be sort of summarized as a generalized reaction diffusion process, where the rate of change of the regional activity is a spontaneous decay, and there is a spatial distribution. The D operator is the spatial diffusion process. And this kind of an equation again can be the solution of this RD reaction diffusion system can be expressed as a linear combination of the kernels. Here, this is e raised to minus beta L, or hi, is the heat kernel. This gives you the spatiotemporal distribution of the activity over this graph, and pi I is parameterized by pi parameters. There are m kernels whose linear combination with mixing coefficients pi I that gives rise to the functional connectivity. So within this model, we assume that we need to learn these parameters pi in order to approximate the observed functional connectivity. And this process is shown here. The left-hand side is the structural connectivity. This is the observed empirical, how regions are connected to each other by physical fibers. On the right-hand side, what you see is observed functional connectivity. What we are trying to do is to bridge this SCFC gap by multiple kernels at different scales of diffusion, local scale, where explains local connectivity, global scale, or a larger value here refers to more global connectivity. And linear combination of this over this structure gives rise to the function. Now the game now is to estimate these coefficients, and you can actually design a minimization procedure for estimating these parameters. What this objective function has two terms. The first term is the typical mean squared error kind of least square error minimization procedure, where x pi to the j is the model, yj is the empirically observed functional connectivity. Now the difference of this is minimized square difference, and over the training set, j is the number of subjects that are available to learn this model. And this is also, there is a regularization term to ensure the sparsity of this model, right, to overcome overfitting and those kind of issues. The L1 regularizer that we used, this kind of regression, lasso regression that we use to estimate these parameters. And this is the these are the results from the blue is the multiple kernel learning model, and the orange is dynamic mean field connectivity model, another kind of model, and the green is single diffusion instead of multiple kernel. If you use only single diffusion kernel, what is the goodness of fit for the test subjects? The goodness of fit here is the predicted functional connectivity matrix, how well it correlates with the observed functional connectivity matrix. You get an average 0.75 or 0.75 kind of fit for with this MKL model. Right, not only that, it fits this, it predicts the functional connectivity matrix, but when we actually look at the networks that are there in this the resultant functional connectivity graph, we see that the communities that are predicted closely matches with what is empirically observed, as compared to very sparse connectivity in single diffusion, or very dense connectivity in the case of dynamic mean field model. Right, this was published in 2018 in scientific reports, and we also have done a couple of other investigations looking at aging and so on. But then I'll move on to a second model, which is so far we looked at the average functional connectivity, but there is a lot of dynamic information that is sort of glossed over in MKL, and we have designed what is called Temporal MKL Model. This is again work of Govinda, our master student who has done this work. Second question that we asked is, can we approximate this functional connectivity better by characterizing the temporal dynamics within the resting state bold time series? And so the assumptions that we made in making this model is that this dynamics can be quantized into discrete brain states, and these states are actually, these states are supported by the anatomical structure. So actually structure constrains this function. Further, we assume that these discrete states lie over a lower dimensional subspace that we can try to uncover this lower dimensional subspace, and then we can propose a solution of predicting this average FC from the structural connectivity by predicting dynamic functional connectivity with the temporal evolution in this lower dimensional brain state space. Right, so I'll make this more clear in the next slides. So essentially what we are doing, instead of looking at average functional connectivity, we look at functional connectivity over windows, and these are called windowed functional connectivities over, let us say, P number of windows. There are different windows over the resting state functional time series, you can now get a series of functional connectivity matrix called WFCs. Now, using these WFCs, right, the training data, say 20 or 30 or 40 subjects from whom we gather all the WFCs and we do a clustering in order to find out what is this lower dimensional subspace that actually neatly summarizes all these windowed functional connectivities observed across subjects. The soft clustering, we used Gaussian mixture model type of clustering. And suppose there are five, just to make this point clear, suppose there are five such states or clusters that exist, then we find out what are the state transitions, how often the brain transitions from S3 to S1 and so on. So that Markov model is estimated along with the steady state sort of distribution of this state sequences. So this is one, and we also, for each of these brain states that we discovered through this clustering, we construct an MKL model for each state. This is called TMKL because now these MKLs now are for distinct temporal brain states. So you learn the same model that we have described earlier, MKL model, but now we have capital K of them over K different brain states. Now, once you learn this model, so the model, again, the learning process uses a lasso type of regression. And once you learn the model, given a test subject's structural connectivity, we can now use this SC to predict what could be the predicted WFCs. So we predict corresponding to these K states, each of them gives you a windowed functional connectivity. We decompose this into a time series. And now you have, let's say, 12 time series corresponding to these 12 different states or 5 different states in this schematic, right? You take those time series and you generate a latent bold time series by looking at this steady state distribution of the states. So you concatenate depending on these the state sequence that you have generated. You concatenate these different WFCs to generate a whole bold time series. And of course, you have to do some thing to stitch these together, some post processing to stitch the time series together. Once you get this bold time series, now you have predicted the resting state functional time series and use this to predict the grand FC, right? Now this is the predicted grand FC, and during learning, this is the difference between this and the actual, the error is used to train these multiple MKL models. But in the case of test, this is the predicted FC. And when we again compare different models, single diffusion, dynamic mean field, multiple kernel, TMKL does better than all of these. And so there is some benefit in characterizing the temporal structure within the functional MRI, resting state functional MRI time series. And we have for this we actually needed to use 12 latent states we discovered, and the connectivity within each of these 12 states is depicted in this sort of a circos plot, which looks at which summarizes state transitions. How often S2, for example, transits to S9 is depicted by a link from these two, and what are the communities in each of these states, and these refer to parts of the 7 resting state networks are distributed among these 12 different latent states that we discovered from the windowed functional connectivity. Now to summarize, this is my last slide. We looked at two questions. One is that how does FC arise from structure? Propose this graph diffusion model, multiple kernel graph diffusion type model, and to characterize the dynamics, we propose TMKL. And there are several questions, many more questions that need to be addressed. For example, how brain networks are reorganized during task. We are now looking at resting state. But during task, how do they become reorganized? And task-based networks kind of dominate, and the default mode is inhibited. All this this process needs to be characterized. And of course, the relation between resting state and task-based activations, and both developmental and aging profile of these RSNs, and then how it gets disrupted in neurodegenerative or other kinds of diseases. So there's work that is progressing along these lines with other students now. We are also looking at graph convolutional neural networks. If you think of as basis for structure function characterization recently. So there are several students who participated in this. Shruti Nayak, who did this dynamic mean field model, is an MS student. She is now in France, Indria, Paris. And Govinda and Joanil were the ones who participated in MKL and TMKL. They are both now doing PhD in University of Maryland. Subbareddy, who has also contributed, is now working on graph convolutional neural networks. He's ready to go to Indria Bordeaux for PhD. And there are two students of mine from University of Hyderabad. The current students, Madhukar, Aditya, Arpita, and Archie, who are continuing this work. We have funding from DST-CSRI to look at dementia, MCI, and also a collaborative project with Srinivas and at IIT Madras and Anirudh from TCS Kolkata, where we are looking at stroke characterization, and other colleagues from IIIT and NBRC. Thank you. Any questions? I'm happy to take.
Sir, good morning, sir. I'm Chandrasekhar.
Yeah.
Sir, here you are using time series, and when we are combining time series before computing the confusion matrix. So, what is the scope of multivariate time series analysis in that particular section? If you perform multivariate time series, is there is scope of improving?
So in the TMKL, you are asking. So this multivariate, this is actually characterizing the multivariate time series using this sort of combination of linear models, right? Linear combination of these MKL models,
Yes,
right?
sir.
That is what we are trying to characterize. Can you elaborate what is?
Sir, in the previous slide, in the last slide.
Let me show you. Okay, the last slide here.
Where you given the block diagram.
Okay, the TMKL block diagram. This
Yes,
one?
sir. Yes, sir. Yes,
Yeah.
sir. Here, in the after eighth step, we are getting time series.
Correct.
Which is
So
where
yeah. So right after the eighth step,
we are getting a time series here, the representation.
Correct.
Where multiple time series are combined. So if you perform multivariate time series, you can find the correlation between the time series, and it can generate good GFC. So
yeah, so this particular time series is a predicted time series based on these are the different time series corresponding to the windowed functional connectivity states that are that are predicted from this MKL model, right? So decomposition of this matrix gives you this time series. Now you have, let's say, 12 of them, but you don't know in what order they are coming. This order state is obtained from this Markov model, right? Which state follows which state and so on. Now, based on this order, we stitch these time series together. Suppose this order is 1 followed by 12 followed by 11 followed by 10. We take the corresponding time series and put it in that place. And of course, at the junction, you need to do some sort of averaging, make this like a continuous time series. So this is
Yes, sir.
the whole bold time series that is generated based on this MKL model. And based on this multivariate time series, I am computing the resultant functional connectivity, predicted functional connectivity matrix. This is compared against ground truth to decide how well the model is performing, right?
Yes, sir. Thank you, sir.
Bapi Raju, can I ask a slightly general question?
Yeah, yeah.
So the thing is, the state transition you're talking about are slightly on a smaller time scale, right? Looks like it's order of seconds.
Yeah, these are typically 20, 30, 30 15 to 30 second millisecond time over the window, basically whatever is that window size.
Yeah, the window size is how long is it?
Actually don't remember now, but
No, roughly,
these are we tried with different ones. The final one is I think 15 this is an 8-minute thing, right? So this is about 15 to 20 second time window.
Okay, no, because what I was wondering is people try to integrate fMRI with EEG and all, right? Now, no, MRI with EEG. The temporal resolution of EEG is combined with the spatial resolution of MRI.
Right.
How do you correlate the fast rhythms of EEG with the slow rhythms of fMRI? This is a general question. Is it possible at all?
General question. Right. Yeah. Okay, so one question is one recent empirical sort of advantage is it's possible to measure simultaneous fMRI and EEG. So to combine the spatial superior spatial resolution of MRI with the temporal resolution of EEG. I've really not looked at, but it's an interesting question. I mean, definitely that there can be a better, if there is a simultaneous measurement, definitely the temporal resolution of these can be improved. So one can talk about microstates instead of these sort of more states which are actually much longer over a period of time. I mean, EEG microstates and how they evolve over time is another thing that can be captured, how they evolve both spatially and temporally. Right. So what is the other question that you're asking is what is the relationship between these faster rhythms slow rhythms that are captured in an fMRI? Yeah, something that we need to look at. I have not really looked at that, but it's an interesting question, definitely, especially in the current context of being able to measure these simultaneously, right? That
right.
People try to use these kind of spectrotemporal decomposition of these slow bold time series, but I've not really seen anything as interesting as one can find in typical EEG recording.
Okay. So there are no more questions. Thanks a lot, Professor Bapi Raju,
Thank you.
for taking time out and coming and joining.
Thank
Thank you very much.
Thank you.